occurs. Infertility affects around 8 to 15% of couples, and in around 50 to 60% of those cases, the male is solely responsible or is responsible in a combined factor. So a male infertility factor is defined by abnormal semen parameters, and it can also be present even when the semen analysis is normal. Now, what are the goals of male evaluation, which have already been covered? Identification and treatment of reversible conditions, lifestyle factors being one of them. Identify any underlying medical problems and serious medical disorders like testicular cancer, pituitary tumor, uncontrolled diabetes, etc. And then finally, detect certain genetic causes which can be treated by ART procedures with the risk of transmission to offspring like Y chromosome microdeletions and all. Now, who are the people who have to get in, who are indicated for a male evaluation? All those couples, male partners of those couples who haven't been able to achieve a pregnancy in 1 year after trial. Earlier evaluation of the male partner is warranted if the man has concerns about his fertility, or if the female partner is more than 35 years old, or if there is any abnormality in the medical history or the physical findings. Now, what are the components of a male evaluation? The initial minimum screening should include a thorough reproductive history and the examination of at least one semen sample. Ideally, 2 samples should be checked with a gap of around 6 to 8 weeks, keeping in mind the time needed for the spermatogenic cycle. Another point to note being previous fertility does not of a new acquired secondary male factor, and the evaluation is the same for men with primary as well as with secondary infertility. Now, coming to semen analysis. Semen analysis is the most important investigation, or it is the cornerstone of laboratory evaluation of a male. It provides important information regarding the volume, so many factors including the volume, concentration, motility, vitality, morphology. All the detailed laboratory protocols and references have been explained and are given by WHO, their latest 5th edition, which came out in 2010. The morphology is evaluated according to Kruger's strict criteria, which was developed in Heidelberg University, this being the main difference of this 5th edition semen analysis WHO protocol from its previous 4th edition. Now, coming to semen collection for semen analysis. All patients should be provided with standardized written instructions, preferably. Abstinence period of 2 to 7 days ideally should be mentioned. In some cases, or in some men with oligospermia, short-term duration of abstinence can benefit them. Semen is ideally conducted by masturbation into a sterile, wide-mouthed, non-toxic container. Coitus interruptus is not a reliable means of semen collection, as the first portion of the semen, which is sperm-rich, can be lost. In selected cases, semen can also be collected after intercourse with the use of special non-toxic condoms, especially in those people who are not able to collect by masturbation, or who are not allowed to masturbate to collect a sample. Actually, we had a couple from Israel, and this Jewish couple, and the male was not, they are not supposed to masturbate and give a sample. So for them, they had to give the sample with the use of a special non-toxic condom. And if the man cannot provide a semen sample, then the post-coital test may give some information according to WHO. Now, the semen should be collected ideally at the clinic. If it is not possible, or if it is collected from outside, it should be kept at normal temperature or room temperature during transport, and it should be examined within 1 hour of collection. If it is being transported in a two-wheeler or a four-wheeler, it should be important should be laid on the fact that it should not be exposed to direct sunlight, it should not be exposed to direct heat, or it should not be kept near the air conditioner of the car. It should be held, it should be covered in a tissue by the person in his hand. That would be the ideal scenario. And following information should be recorded in the form, or as well as on the sample collection container. 2 unique identification numbers should be there, including the man's name, date of birth, and the hospital number. You can use any 2 of them minimum. And semen analysis should start within the first hour of collection of the sample. In the first 5 minutes, as soon as you get the sample, you have to keep the container in an incubator at 37 degrees and wait for liquefaction to occur. Normally, liquefaction occurs within 30 minutes. You can wait up to 1 hour in certain situations. And between 30 minutes and 1 hour, as soon as the liquefaction occurs, then you start assessing for liquefaction, the appearance of the sperm, appearance of the semen, measure the semen volume as well as measure the semen pH. Now, what are we evaluating macroscopically? What all can we see and we can evaluate? First of all, we'll evaluate the color being gray white and the appearance being opalescent and turbid. Opalescence is due to the presence of the proteins which are present, and turbidity is due to the presence of the sperms which are present. Liquefaction, which usually is supposed to occur within 30 minutes, but you can wait up to 1 hour in some cases. The semen volume is then measured, and the lower reference limit is given by WHO 5th edition as 1.5 ml. The pH is measured using a pH paper. It should be more than 7.2. And viscosity, which is the ability to fall freely as drops from a pipette, or threads which are formed are less than 2 centimeter in length. This shows a normal viscosity. And coming to the details, the color is usually gray white, and in case of presence of RBCs or hematospermia, the color can be red. If urine is present, it can be light yellow. If bilirubin is present, it can be orangish in color. Normal liquefaction, as we mentioned, is within 30 minutes. If there is a delayed liquefaction for more than 60 minutes or 1 hour, then you can take any of the 3 steps by adding a physiological medium, almost equal volume, like double-glucose phosphate buffered saline, or you can use a mechanical method like repeated pipetting or syringing through gauge 19 or 18 needle, or you can try digestion by bromelain, which is a proteolytic enzyme. And the pH, which is normally more than 7.2, can be increased due to acute infections, especially those affecting the prostate or epididymis, or due to delayed examination also pH. Now coming to the analysis of the volume. The normal range or volume is kept as 2 to 5 ml. Aspermia is the absence of ejaculation during an orgasm. When the volume is less than 1.5 ml, it is called hypospermia, and it can be due to decreased abstinence period, due to spillage, due to obstruction, due to partial retrograde ejaculation, or due to any accessory gland malfunction. The term hyperspermia is used when the volume is more than 5 ml, and it can be due to some causes like infection or due to prolonged abstinence. In some cases, it can be due to contamination with urine also. Now coming to the detailed microscopic analysis, a wet preparation is prepared, and for that 10 microliter of semen is spread on the maclar or Neuber counting chamber, and it is covered by a cover slip of 20 to 22 micrometers, so that the depth of the semen column is around 20 micrometers, which would be ideal for assessing most of the parameters. Now, what are the microscopic parameters which we are going to look at? We will be looking at the motility, which should have a total motility of around 40% according to the latest criteria. The progressive motility should be 32 or above. The amount of live sperms or vitality should be 58% and above. The sperm number means the total number of sperms present in an ejaculated, and that ejaculate sperm number which is needed to be normal is 39 million in per ejaculate. The sperm concentration, which is more than 15 million per ml, and the morphologically normal sperm should be more than 4%, and the number of WBCs present should be less than 1 million per ml. Now, what are the methods for assessing the motility? It can be manual assessment or by CASA. It should be assessed as soon as liquefaction occurs, preferably at 30 minutes or within 1 hour when as soon as ejaculation occurs, because as time goes on, it can change due to the changes which occur in the semen sample due to dehydration, due to pH, or due to changes in temperature. You have to start scoring a given field randomly, and you have to count at least 20 intact spermatozoa. Avoid motile pinheads, and they should not be counted according to WHO. Now, if the progressive motility is less than 32% or the total motility is less than 40%, it is termed as asthenospermia. But the simplest system for grading motility has been given by WHO. A progressive motility has a variety from the earlier staging system. Now it is only 3 categories. Progressive motility, when spermatozoa are moving actively either in a large circle or in a linear fashion. Non-progressive motility, all other patterns of motility, whether they are moving in a small circle or not. And immotile, when there is no movement. The clinical significance of this is that the presence of a large proportion of vital sperms without motility may be indicative of structural defects in the flagellum, whereas a high percentage of necrospermic sperms may indicate the epididymal pathology. Now coming to the count, the counting chambers normally used are macular, Neuber or microcell. Alternatively, a hemocytometer can also be used, which is cheaper when compared to all these costly foreign ones. And the concentration of spermatozoa is then calculated according to the formula. The minimum count which is required is more than 15 million per ml. When the count is less than 15 million per ml, it is termed as oligozoospermia. And azoospermia, which is seen in around 1% of males, is the condition in which no sperms are seen in the ejaculate. It might be due to testicular failure or it can be due to an outflow tract obstruction. But the diagnosis of azoospermia can only be confirmed after centrifugation of the sample at 3000 G for 15 minutes and examination of the pellet. If still no sperms are seen, then you can term the person as azoospermia if 2 samples have been checked. Or if you are seeing sperms after centrifugation, it is termed as cryptozoospermia. Now what to do in the next 3 hours? You have to send samples to the microbiology lab if it is needed, and then fixing and staining the smears for morphology has to be done. And later in the same day, subsequent tests have to be done. This is from the WHO manual. So what are the stains which are using for morphology? Pap stain, Shore or Diffuse stain can be used. A multi-key counter can be used with one for normal ones, one for abnormal, and one for each of the 4 abnormalities like head, mid-piece, principal piece, and the cytoplasmic droplet or excessive cytoplasmic reticulum. Because free tails are not counted, pinheads should not be counted as sperms at all. Now, what are coming into a little bit of the details regarding the type of defects which we are expecting in the head, the net piece, the principal piece, as well as the excess residual cytoplasm. As you know, the size of the head should be between 3 to 5 micrometers and all. The presence of more than 2 vacuoles covering more than 20% is abnormal. The chromosomal cap should be covering 40 to 70% of the area. And the other details you can go through by yourself. But another important point to note being that spermatozoa which have that irregularly stained cytoplasm, which are 1/3 or more of the size of the head, are considered abnormal. And this abnormal excessive cytoplasm should not be called a cytoplasmic droplet. It should be termed excess residual cytoplasm. Now, this is a picture showing some of the abnormal forms of human spermatosoma. As you all know, there are some affecting the head, some affecting the neck and midpiece and tail, as well as excess cytoplasm. Now, coming to the other cells which are present in semen along with the sperms, some of them may indicate a damage like immature germ cells, some may indicate a pathology, or some may show an inflammation like leukocytes. So when the total number of round cells exceed around 1 million per ml, then you have to do the peroxidase test and find out whether that number of WBCs is more than 1 million or not. Normally itself, a particular number of round cells are present. When the concentration exceeds 1 million per ml only, we have to get alarmed. Now, another information which we have to decode from this semen analysis is the total motile sperm count, which is calculated by multiplying the volume of the semen sample with the concentration of the sample with the percentage of total motility, which will give us the total motile sperm count. For example, if the count is 10 million and the volume is 4 ml and 50% motility, then the total motile sperm count will be around 20 million, which will give us a rough idea as to how the treatment should be targeted. If the total motile sperm count pre-wash is less than 1 million, the literature advocates ICSI. When it is between 1 to 5 million, IVF can be an option. And when the pre-wash TMSC is more than 5 million, IUA can be an option. So what are the minimum required for doing semen analysis in a standard setting? There should be an in-house semen collection room. Masturbation is always the preferred method. Analysis has to start within 1 hour of ejaculation. The minimum abstinence period of 2 to 5 days have to be followed. Well, don't forget the universal precautions you have to take and the well-trained personnel you have to have in a standard accredited laboratory. Now, we can decide on further testing also depending on the semen analysis results. For example, endocrine evaluation is indicated in men if their semen concentration is less than 10 million per ml. Genetic evaluation is indicated in men with severe oligospermia, for example, less than 5 million per ml. These are from ASRM Practice Committee Guideline 2015. Now, we can give serious consideration for ICSI if the sperm count is less than 5 million per ml or if the progressive motility is less than 10% with a normal morphology less than 4%. So this is according to Godwin. Now, as we have covered semen analysis in basic detail, we'll come to the sperm function test. To know what are the sperm functions we are testing, we should know how the sperm travels. First, whether they are alive and motile, whether they are able to traverse the cervical mucus and the uterus and undergo capacitation followed by acrosome reaction, then zona binding, penetration and ovum penetration. So vitality tests, capacitation tests, acrosome reaction tests and zona binding and ovum penetration tests are the few of them, followed by special tests like DNA fragmentation, ROS and antioxidant capacity, and last but not the least anti-sperm antibody tests. So these are indicated not in everybody. It is not a routine test which is given for all males who are attending the fertility OPD. It is used especially when there is a repeated IUA or IVF failure and there is an advanced male age of more than 40 to 45 and there is smoking and other lifestyle factors are present, when there is a previous low fertilization and when there is varicose with normozoospermia. These are the special cases where we plan for these tests. So coming to the sperm viability test, viability tests are used especially when the motility is less than 58%. There are 3 types of tests are useful. A dye exclusion test using trypan blue or eosin-nigrodin. The sperm cells which are stained dark are dead and cannot be used, whereas the viable sperms will be lighter stained. But the problem with the dye test is that these viable stains are also cannot be used after the test. So we'll be using more of hypoosmotic swelling test in which there will be swelling or curling of the tail of a viable sperm, and these sperms can be selected and used in ICSI. Another novel method is the use of being exposed to pentoxyphilin. Viable sperms will start having motility after a certain time. Now coming to capacitation test, capacitation being a very important stage when the sperm undergoes cell surface changes as well as achieves hypermotility. The tests are, first of all, sperm wash with incubation albumin containing solution and identify the changes manually or by computer-assisted semen analysis. The second one is a staining test with the chloride tetracycline, and there's a specific staining pattern which can be identified by fluorescence microscopy. It is shown by capacitated sperm. Now coming to acrosome reaction test, there has been a strong relationship between the normal sperm morphology and its inducibility of acrosome reaction as shown by Dr. Daniel Franken et al., who along with Kruger made the Kruger's criteria. These normal samples demonstrate spontaneous acrosomal reaction of less than 5% and induced reactions of more than 14 to 40%. The inducers used are calcium ionophore, follicular fluid, and steroids. The techniques used vary, and some of them are those involving fluorescent lecithins and flow cytometry. Now coming to sperm DNA fragmentation, the sperm DNA damage has effects on fertilization, embryo development, as well as on pregnancy. The intrinsic factors like protamine deficiency or mutations can be the causative agent, or it can be due to extrinsic factors like heat, radiation, and gonadotoxins. There are several methods which can be used. The commonest ones are being outlined here. The commonest ones being sperm chromatin structure assay, which is a CSA, which is a standardized method. Tunnel, nick and labeling. Double-stranded DNA damage can be detected. Single gel electrophoresis. It is done per sperm. It is known as COMET. And one of the simplest methods, sperm chromatin dispersion, which measures the absence or presence of halo, and which is shown in the picture below. That is very user-friendly test. Now, what is the association between sperm DNA fragmentation index and IUI? As we can see, even though the clinical pregnancy rates are not that much decreased in the presence of DFA more than 30, the early abortion rates are significantly higher in those populations with DFA more than 30%, and it comes down with low sperm DNA fragmentation index. Now, what is the association with the ICSI? In ICSI, pregnancy rate was significantly decreased in those with a high DNA fragmentation index, as shown in that human reproduction study which came out last year. Now, coming to sperm aneuploidy assay, FISH is the technique which is used to do fluorescence in situ hybridization. Typically, around 5 to 7 chromosomes of an interface sperm nuclei are checked, especially which is done only in at-risk individuals like those with a translocations or those with severe oligozoospermia or teratozoospermia. This is done especially keeping in mind that aneuploidy can negatively influence the pregnancy as well as delivery rates. As you can see in this picture, if there is a damage to the DNA, if the damage has been repaired, it can result in a normal offspring. But if there is no repair, then there might be a fertilization failure. And if there is a partial DNA repair, then it can result in a fertilization and it can result in an abnormal offspring, as you can see. So that is the importance. So coming to semen reactive oxidant species test, several techniques like chemiluminescence and flow cytometry are used, but their clinical value is unclear, and treatment with antioxidant seems to be a very reasonable option. The total antioxidant capacity is another one of the tests. Their measurement is also done using TRAP or task, total radical trapping antioxidant parameter, or total antioxidant scavenging capacity. The methods used being chemiluminescence, photochemiluminescence, and calorimetric assay, but their clinical value is still unclear. Now, coming to anti-sperm antibody test. Anti-sperm antibodies are suspected when there is extensive agglutination present. That means sperms, motile sperms sticking to other motile sperms, head to head or tail to tail or head to tail. The risk factor increases with trauma, torsion, previous vasectomy and all. There are qualitative tests like MAR, mixed agglutination reaction test, and there are quantitative tests like ELISA. The clinical utility is uncertain, especially in the case whether ICSI is planned. Another important new study which has come out in the recent times is that if ASA is present in sperm, then there is no need to do a diagnostic testicular biopsy. Now, what are the take-home baby messages? Meticulous semen analysis in a standardized laboratory is a must. If there is a doubt or bad result, then repeat after 6 to 8 weeks before labeling that man infertile. Lifestyle factors need to be repeatedly addressed as they have a very vital role. The sperm function tests are not recommended as routine use diagnostic tests, and the role of sperm function tests in ICSI is controversial because of the fact that there are two schools of thought. One school of thought saying that because only one sperm is needed for ICSI, why test? And the other school of thought saying that one sperm is still needed for ICSI, so why not do it?